So please tell us about this anti-venom drug.
It's a peptide which is basically a very small part of a protein. So it's very small in size, and that makes it stable, and our data suggests that it is quite effective in neutralizing the snake venom. So if somebody has a snake bite and they take injection of this peptide, then it effectively neutralizes the effect of the venom and the patient can be saved.
So sir, like, can you mention in brief, like, how many cases of snake bites are there in India? Approximately something.
Yeah, I mean, I think approximately my guess is several, like something like 10-20 lakhs probably people get snake bites every year, and more than a lakh die of snake bites. That I know, because that number I have seen in the literature. So about a lakh, more than a lakh basically die in India, more than a million die across the world.
Okay. So sir, can you please tell us in detail the science behind it, how this drug was made?
So the discovery of the fact that this peptide is effective in neutralizing the peptide, I mean neutralizing the snake venom, is 20-30-year-old discovery. So that's not a new thing, but the concern was that normally you would make this using synthetic processes, where the cost of the peptide is significantly high. So the cost would be a problem. So what we have done is we have found a way to manufacture this peptide using recombinant DNA technology, and that allows us to manufacture in a much more cost effective manner. Because if you look at from an application perspective, most people who would need treatment for snake bites are people who are living in rural areas, and generally these are poor people. So for them, not just the availability of the treatment, but then also the cost of the treatment is equally important.
Okay. So can you mention some of the steps, like how the research process was done?
Okay, so the discovery that this peptide works, that was done by actually an Indian origin researcher based in US. That was 20-30 years ago. Then the idea of using this peptide and developing in a recombinant DNA technology, that came up when we had a discussion. We actually had a visitor here from San Jose University. There was a professor, and she and we together basically had the plan to address this. She is the one who actually did the genetic manipulation of the E. coli to manufacture or to make this peptide, and then our lab took the task of optimizing the process, purifying it, characterizing it, and all that. So in a way, it's a joint collaboration between IIT Delhi and San Jose University.
Okay. So sir, how this new drug is different from the other ones that are already there in the market?
So the treatment that is available today is basically using horse serum. So the way it's done is that you take the venom from the snake, you inject it in the horse, and horses are known to be immune to snake bite. So basically what happens is that as soon as the venom goes into the blood of the horse will start producing antibodies. Okay. So then what they do is they take the blood serum from the horse, which contains these antibodies, and that is stored and then used as the therapy. So that's what we do today. The problem with this whole process is that besides the fact that of course it's painful for the snake, painful for the horse, but outside that there are two major problems. One is that the serum that you get is has limited stability. So it has to be stored in the cold, and it can only last anywhere like 6 months to 8 months time, right. So it has limited shelf life. It needs a particular storage, which again restricts it into larger hospitals. So normally this is not something that you will find in villages or small towns. So if anybody has a snake bite, they have to rush the person to the to any major hospital close by, and generally what happens is that because of this delay in treatment, even if the patient is saved, they are going to have some major major injury or major repercussions because of the snake bite, because of the delay. So that's a big problem. Second problem is the cost. So the cost of this treatment is anywhere around 5,000 to 10,000 rupees per dose. So, that cost again is quite high for someone who is poor or living in a rural area. So, these two are the major kind of problems with the present treatment.
So what is the cost of this new drug?
So, again we have that is what our lab has been working on. So, the process that we have now, we are expecting one dose to cost around 60-70 rupees. So, it will be substantially smaller than the cost of the existing treatment.
And how many doses are recommended?
Generally, it's single dose. So, you give a dose and then if it works, then it will neutralize the snake venom. So, it's not something you would need multiple doses for.
Okay, so like if a person has been bitten by a snake and the treatment is given after 2 to 3 hours, will the drug still be effective?
It will be effective because as far as the venom is in the blood of the patient, so it will neutralize that, but whatever harm has already happened, the treatment will not be able to reverse that. So, it can only basically stop anything further from happening, but whatever has happened has happened.
Okay, so are there any side effects also of this drug?
So, again this drug we have not taken it to human testing yet, right. So, it is early to say what the side effects would be, but we are obviously hoping that there would not be any major side effects. But that is actually another point, that with existing treatments there is a potential of side effects because you are injecting blood serum. So, a lot of people have additional side effects because the serum contains not just the antibodies that you want, but it will also have many other things in it, right. So, a lot of patients have reactions and stuff like that to the existing therapies. So, that is a problem with actually current therapies, but we are hoping that it would not be the case with this thing, because ultimately this is it's a peptide. So, it is a relatively very small molecule, right. So, we are hoping that should not create any major reactions.
Okay, so what was your research models like, on whom you tested this drug?
Okay, so far we have tested it against the venom of the Russell viper and the rattlesnake, and then against those snake venoms at least. So, against those venoms the peptide that we have made seems to be quite effective. Now, the next step for us is to test it against more snake venoms, particularly those that are commonly found in India. And that is so we are hoping to see that the peptide works against most of the snakes at least, and once we see that, then the next step would be to work together with a major company to take it forward and test in humans and get it commercialized.
Okay, so can you mention some of the snake species which are like useful for this type of snake bites?
I don't. सर रोक लो एक मिनट है ना, रोक लो, लिखना पड़ेगा क्या क्या भैया. The 4 major snakes that are often studied for India include the cobra, the Russell viper, the saw scale viper and the crate. So, these 4 we would like to test in the next round.
Okay, so like by when you are expecting this drug to be in the mainstream market?
So, the process of approval of the drug is of any drug actually is a fairly long process. So, it is difficult to say by when it will come to market, but for example, testing against these snakes I would expect to be finished in the next few months. Okay. Then finding the right partner for commercialization, I am hoping we should be done by end of the year. But then thereafter it may take, normally it takes anywhere from I would say a year to 2 years to get all the approvals that you would need for the drug. So, at least couple of years down the road.
Okay, so can you mention some of the major errors while you were carrying out this research during the trial and error process? What were the major errors that you encountered?
I don't think that's a good question for us. We didn't encounter any major errors. Yeah.
So, sir, apart from all this, would you like to add anything else which you think we have missed around?
No, I mean, so I think like I said, our goal is to focus on this project and deliver this to the markets as soon as we can. And I mean from the process part of what we have done here, we are confident of that part, but the issue here is obviously that human biology is fairly complex, and so the real test will be when we do these next round of studies in rats, and then followed by the real test will be actually the human studies, right. So, till then I think at least we are keeping our fingers crossed, hoping that those results turn out to be well, and I we sincerely hope that this drug reaches the market. Okay.
Thank you so much.
Thank you.